In my last presentation, we looked at the research done by Dr. Bana Jabri. Dr. Jabri worked clinically as a doctor in pediatric gastroenterology. She is currently teaching and doing research at the University of Chicago Department of Medicine, with a focus in celiac disease, irritable bowel disease, and other immune disorders that impact gut health. As a recap from last time, we learned that celiac disease is an inflammatory disease of the small intestine that is triggered from the ingestion of gluten protein, found in wheat, barley, oat, and rye. In the previous paper, Dr. Jabri developed the first pathophysiological animal model for celiac disease. She determined the key effector cells that drive tissue destruction, otherwise known as villus atrophy, in a mouse model. Looking at the graphic, we see that TG2 increases the affinity for gluten peptides, for the HLA DQ2 and DQ8 haplotypes. These haplotypes are genetically susceptible in terms of celiac disease, and the antigen-presenting cell produces IL-15, which indicates active celiac disease. IL-15 plays a key role in epithelial cell damage, which we know as villus atrophy. This paper was published February 12, 2020. Now, we're going to look at previous work that led to Dr. Jabri's novel animal model for celiac disease. Chronic inflammation permanently reshapes tissue-resident immunity in celiac disease. This paper was published February 7, 2019, almost exactly a year prior. It was known that celiac disease results in chronic inflammation of the small intestine in the absence of a gluten-free diet. But it was unclear how the lymphocytes that reside in those tissues respond to chronic inflammation, and if that chronic inflammation can permanently change the T-cell compartment of those lymphocytes. In this study, patients were classified into 4 groups. The control group was made up of patients who had symptoms of upper gastrointestinal tract disease, but no duodenal inflammation or a family history of celiac disease. They also had a negative serology for TG2, which is that deamidating enzyme in the pathway for active celiac disease. Another group was isolated with active celiac disease, indicated by villus atrophy and a positive serology for TG2. The third group were patients who have celiac disease but were treated with the gluten-free diet. There was another group that contained patients who were positive for TG2 but had no histological symptoms. In Figure 5a, a T-cell receptor gamma variable protein-coding gene was used to characterize all expressed TRG and TRD gene rearrangements in V delta 1 positive T-cell populations. Figure 5 shows the proportion of unique CDR3s, complementary determining region of the heavy chain, where the rearranged gene segments are joined. This is where DH joins VH and JH. Using a specific TRGV for the V delta 1 positive peripheral blood lymphocytes and intraepithelial lymphocytes in the control, active, and gluten-free diet patient groups. There was a significant difference seen between the control and active celiac groups' peripheral blood lymphocytes using the TRGV2. For the intraepithelial lymphocytes, a significant difference was also seen between the control and active celiac group when given TRGV3. The most significant finding was seen between the group's intraepithelial lymphocytes when given TRGV4. The control group showed a higher proportion of unique CDR3s compared to both the active celiac and gluten-free diet groups. The loss of TRGV4 gene transcripts in the active and gluten-free diet group indicates the switch from innate-like intraepithelial lymphocytes in their healthy state to interferon gamma-producing intraepithelial lymphocytes in celiac disease due to cellular turnover. The next paper by Dr. Jabri, identification of a YC receptor antagonist that prevents reprogramming of human tissue-resident cytotoxic T cells by IL-15 and IL-21, aims to determine whether BNZ2 can block the gamma chain cytokine specifically, and the effects that BNZ2 has on the intraepithelial cytotoxic T cells. The graphical abstract depicts the process of IL-15 and IL-21 induced proliferation and cytolytic activity of the immune cells in the intestinal tissues. This is important because celiac disease is marked by the upregulation of IL-15 with an anti-gluten CD4 positive T-cell response, resulting in the tissue destruction, as well as the IL-21 enhancement of the adaptive immune response and interferon production. Using the same format for patient groupings as the last experiment, samples of the duodenum were used for analysis. Figure 3C shows density plots indicating the effect of BNZ2, which is shown in blue, on the IL-15 and IL-21 induced gene expression changes. Those are shown in red. On each density plot, there is a histogram indicating the number of differentially expressed genes, or known as DEGs. The number of differentially expressed genes for all three plots is greatly decreased between the cytokine and cytokine plus BNZ2 groups. This shows that BNZ2 not only prevented IL-15 and IL-21 mediated transcriptional alterations in the IECTLs, but also prevented those alterations in the small intestinal epithelial compartment, which includes both IECTLs and epithelial cells. So what is the big picture takeaway from these two experiments? First, Dr. Jabri discovered that chronic inflammation can permanently change the intraepithelial cells that reside in the tissues of the small intestine through cellular turnover. Secondly, Dr. Jabri found evidence that BNZ2 can efficiently prevent the transcriptional alterations caused by the upregulation of IL-15 and IL-21 in active celiac disease. These two experiments highlighted the molecular changes celiac disease-induced chronic inflammation can make to intestinal immune cells, and a gamma chain receptor antagonist that can prevent the previously mentioned reprogramming of those cells. This last slide shows a quick timeline of Dr. Jabri's research thus far. First, she determined how lymphocytes in the small intestine are altered by chronic inflammation. Next, she determined BNZ2 as a receptor antagonist that blocks IL-15 and IL-21 production. Most recently, she's determined the first pathophysiological mouse model for celiac disease in humans. The circles in the top right of those squares show the order in which I have presented them.